All right, I'm going to start on the chapter on inheritance here. Um, there's a lot of slides. I'm going to go fast, so you can stop at any time. And some of this stuff is less than crucial. And so, what we really want to discuss is how plants were used to solve the puzzle of inheritance. And that was done by a guy named Gregor Mendel to describe how genes work. And we'll go over maybe a simple genetic problem or two. Mendel's often considered a genius, and the book kind of presents him as solving this really important problem kind of out of nowhere. And I often um, they often present the breakthroughs as the work of a genius, and I really don't like that. I don't think there's many people who are geniuses. And one guy that they used to present as a genius is Elon Musk. They don't anymore because he's been in the press more. Um, but I think often geniuses are products of their time. And so while he seemed like a genius when he became the first guy with an electric car company, and he had a solar company, and now he runs SpaceX, which is a rocket company, it seems like, man, he's uh, really something smarter than the average guy. But really, when you look at it, he's a really wealthy guy that invested in businesses, not an engineer, didn't invent these cars, didn't invent space travel, but he bought companies and he ran them pretty ruthlessly, but and had some success. But if you look at another guy that had a similar career, Jeff Bezos, it's kind of the same thing. Both of these guys started as internet entrepreneurs. They ran early companies that did extremely well by being among the first on the internet. For Elon Musk, it was PayPal, one of the companies that figured out how to get money on the internet. And for Jeff Bezos, it was Amazon.com. Both these guys have successful rocket companies. And I think when you talk about Mendel, it's kind of the same thing. He was a product of his time. Um, Inheritance seemed like a puzzle that was too difficult to solve. How could all the information needed to make a person or a plant or an animal, how could you store that in a single cell, like a sperm cell or pollen cell? Um, Biologists couldn't figure it out, and they were focused on animals and people, and so they thought about fluids, and inheritance just seemed too complex to understand because they were focused on traits like intelligent or size and things like that. Um, but there were people that were doing applied work on inheritance that were making lots of progress. And these were farmers. They were people developing new techniques for the selective breeding of plants and animals. They included these guys here on the bottom, Robert Bakewell and Thomas Cope. And they encouraged people who were farming to keep really good records and to use selective breeding to improve the quality of their plants and animals. And just as an example of how this was done, it was you would you would you would breed the best animals together. You would keep a record of, like, if you were a dairy farmer, you'd keep a record of how much milk cows were producing, or which sheep had the best fleece, and you would make sure that those sheep would breed, that the females would breed and not females that weren't producing as much. Here's a good example of the results of selective breeding. This sheep on the top there is a sheep from Australia that escaped and was living in the wild for a little over a year. Obviously, when they're living in the wild like that, they can't have their fleece removed. And so that wool accumulates over time. And that fleece there weighed over 200 pounds. That's the wool that the sheep has. And so these were selectively bred for wool production. And when that sheep was recaptured and sheared, it looked like this normal sheep. No wild sheep would ever produce wool like that. It's only through selective breeding. And so Mendel used principles of selective breeding and very good record keeping, and he worked with plants. And plants were really nice to work with because he could control their reproduction by controlling what happened to the pollen and closing off the flowers. And he selected traits that were really easy to follow and were didn't show any blending or intermediates. Things like flower color, um, flower position, seed color, seed coat. And then using plants, he could run through 3 generations each year and produce hundreds of offspring. So he could see patterns that were kind of large scale. Whereas if you were studying the breeding of cows or sheep or people, gener each generation took more than a year to mature. And with people, it was 20 years. And so that was really a big thing, was choosing the right thing. He could run experiments. And so he could see that fluids weren't involved. Traits were passed down from each parent, um, the parent that supplied the pollen or the parent that supplied the egg cell, and that these were independent of one another. Okay. The approach he used was the test cross, where he would use true breeding parents that would always produce, like in this case, flowers of the same color, and he would cross them. So he would have true breeding purple flowered plants bred with true breeding white flowered plants. And when he did this, he found that the offspring were always purple. And so he called the purple the dominant trait and the white the recessive trait. And he could do these crosses because he could do pollination by hand. So he controlled fertilization and he could produce many, many offspring. When he allowed these hybrid plants that had both purple and white parents to reproduce, he found that he always got the ratio of 3 purple to 1 white flowered offspring. Okay, and he did this with many different plant traits, and he found that the ratio was always 3 to 1. And so this counting was really important for him, and producing large numbers of offspring was really important. And he followed 7 traits: flower color, seed texture, pod color, pod shape, flower position, and plant height. And he found that this worked for each of them. So he concluded that traits were transmitted by discrete factors. We now call those genes. Each individual receives 1 copy of a gene from each parent. That's why you have 2 parents involved. And not all copies of the gene were identical. We refer to them as alleles, alternative forms of the allele. So he distinguished what the genes were present, what genes were present from the physical traits. You could have 2 of the same alleles and be homozygous, okay, and then you would be true breeding and you would show what that allele was. But if you had different alleles, one of each type, you were referred to as heterozygous, and one of those alleles could be hidden, okay. Although it was hidden, the allele would remain discrete, and so presence of an allele doesn't guarantee expression. Dominant alleles were expressed, but recessive alleles could be hidden. So he distinguished genotype from phenotype. So these were his big advances, and this couldn't explain everything, but it could start to explain some things from inheritance. So this test cross was a really powerful approach that proved that genetic traits were discrete or particulate, not blending, and that dominant traits could hide recessive traits. So this was really, really important. This was our first application or first kind of explanation of inheritance. But of course, this came from agriculture. They knew this worked because they'd been doing this for hundreds of years. Um, and Mendel selected true breeding pea strains because he knew this. He was raised on a farm and he studied probability in school, and he knew he knew what he was doing. So this F1 generation that where he was crossing the true breeding strains, always the dominant trait was always expressed, and the recessive trait was always hidden. No, he chose traits that had no intermediate characteristics so that he could kind of show this pattern. In his second generation, where he allowed these hybrid plants to self-fertilize, the hidden traits reappeared, okay, in that 3 to 1 ratio. Okay, but it's really that 1 that you see is a homozygous recessive, and the 3 is actually some plants that are homozygous for the dominant trait and some plants that are heterozygous. Okay, and so this discovery of the ratio is A 3-to-1 ratio is actually 1:2 to 1. Okay, and we now express this, whoops, with looking at what are called Punnett squares. And so the purple plant had 2 copies of the dominant gene. The white plant had 2 copies of the recessive gene. The recessive gene is shown in lowercase, the dominant gene is shown in uppercase, and each parent can only pass on one trait through pollen or egg. And so we see that the dominant gene is going to be made from the purple parent. All their allele or all their um, pollen or eggs are going to carry that dominant gene. And so all the offspring will have one dominant and one recessive gene. Then when the F2 cross is made, we have individuals that can make either type of gamete, one with a dominant gene or one with a recessive gene. And this is where we get our 3-to-1 ratio, which is 1:2 to 1. So that's how we would solve that. I'm going to skip the F2 generation and so talk maybe a little bit about advanced genetics. That in agriculture, we know some traits that we can't map like this. We could see lots of applications of traits that we could map with Mendel's, but we could see that there were clearly some things that were linked to sex, like hemophilia. Um, so here we have hemophilia almost always occurred in boys and not in girls, and that's because it was sex linked. It was happening on the X chromosome. We'll talk about these more advanced things in the next lecture.